Уважаемые коллеги,
Distinguished colleagues, thank you for coming to our session. We are going to be discussing the same topic that we started this morning. Many of you have attended the plenary session, which was an excellent, really excellent event with lots of interesting presentations and stories of digital medicine and new drugs being created. When we talk about HIV infection, we could continue discussing to follow up on what we discussed in the morning and the plenary session with Mayor of Moscow and Minister of Healthcare of Russia, and they discussed pretty much the same ideas that interest us today. Now let me give the floor for moderating our session to Mr. Alexander Melerzanov, who will be moderating our discussion, and he will explain to us why. Good afternoon, dear colleagues. Thank you for introducing me. It is true, it's a good question why a dean of Moscow Institute of Physics and Technology moderates a discussion on a medical topic. You are all aware of the fact that healthcare and medicine have really changed in the past 20 years. And the medicine of today is done primarily by scientists, and doctors use the achievements of science and ask questions. Mathematicians, physicists, biologists do the rest. About 10 years ago, at our Moscow Institute of Physics and Technology, we started a new program. We were actually established about 70 years ago. We have always been an institute or university of technology, and we always worked under the government and worked for the needs of the government. A few years ago, we understood how medicine needed mathematics and physics, and we established this new faculty of biological and medical physics. We set up a special entity for healthcare professionals, but they are not medical doctors. They study mathematics and physics, IT, and English, because they really need to know English if they want to be part of this system. Some of these people, some of the students, have this dual course with Sechenov University, and they get two diplomas actually. This is our pilot project, that they get a diploma of a physicist and a diploma of a physician, and that's how we started our program. Scientists and doctors often misunderstand each other. They have different backgrounds, they have different terminology, and here we have a new system where doctors and scientists study together for the first 3 years. In 2018, we had our first graduates, and today we see the results of that. We see new inventions and new patents and how this whole area is being developed. That's to answer your question why I moderate a medical session, being a dean of a physics university. Medicine is a high-tech sphere today. We need new medical materials and devices, vaccines and drugs. Many of those things are invented in scientific laboratories, including new drugs that are designed using computers, and they need to know computers very well and need to have some technical education. Let me give you an example of how physicists are involved in medicine today. and they understand what it is, and they get pretty high, we will show you a video address of Sir Konstantin Novosolov. He is a graduate of our university and a Nobel Prize winner. In the 21st century, we discover new technologies for living systems. Academy member Ginsburg, in his article on the physical minimum of the 21st century, he describes what we are to expect from physics in the 21st century, has written that biology using more advanced physical methods has been progressed rapidly after genome sequencing and now is in the lead in science. Konstantin Novosolov is another great physicist and a Nobel Prize winner, whether it's true today, and what kind of discoveries are we to expect in the nearest decades. Gene sequencing is important, and that really turns biology and chemistry into highly attractive and fast-growing disciplines of today. The main problem of biology and medicine still remain. They study highly complicated systems, human bodies, and we don't have enough terminology to describe the whole complexity of our bodies. So I would say that we need to expect breakthroughs here, and these breakthroughs will be at the border between genetics, biology, mathematics, and physics. When new methods of describing complicated systems, and they cannot come from within biology. They will come from outside of biology, where these disciplines overlap due to this link between physicists, mathematicians, experts in genetics and biology. And that's a promotion of tomorrow's session on new drugs as part of this conference in pavilion number 75, Sklifosovsky Hall. Moving on, also our University of Physics and Technology has been developing quite rapidly in medicine. We've been developing our international cooperation, and we are in cooperation with People's Republic of China and some of its leading universities. We have exchange a bit of our professors and students, and we have been developing our joint projects. We really hope that we will have a joint program on HIV vaccine. We are aware of the fact that this is an important topic, and altogether we need to understand that HIV infection is a comprehensive problem that we need to tackle from all these different sides. Later we will discuss the importance of genetics and anything that has to do with genetic features of people who are carriers but they don't get AIDS. And HIV and AIDS are not the same things, and acquired immunodeficiency syndrome is one of the results of like a clinical manifestation of HIV infection, and sometimes you can be a carrier of HIV without clinical manifestations, and sometimes we need to understand why it's like that. Here in Moscow, we have achieved significant progress in fighting HIV infection. We can be quite proud of that result. And Alexei Mazus is an expert in that. He can tell us why we're so successful and how we can start developing it. Thanks a lot. We've had quite a productive morning. We have shown some slides comparing Moscow and other capitals of the world. We have shown the differences between cohorts of patients and Moscow, New York, and Europe, we had a really detailed discussion. We have different patients, different cohorts of patients. There are some similarities, but some things are really different. We're having issues with the clicker again. They brought a larger clicker, but still we really had issues with it in the morning. All right, here's one of the slides that we showed this morning. What's happening with HIV infection? Can we achieve the ambitious goals of the UN AIDS program and the WHO to eradicate HIV by 2030? There's this 1990 program, and they believe if you implement that program, this will be the end of HIV, and there are measures that we need to take. They need the WHO recommends to do circumcision, to use condoms, to use antiviral drugs, and treat HIV to suppress the viral load. That's a pretty clear position. In 2011 in Baltimore, we discussed that particular article, which said that if we treated the maximum amount of HIV patients, the epidemic will be over. They chose a particular location, and they would just treat everyone with antiviral drugs as soon as the infection was diagnosed. They started treating, and mathematically it was quite simple. If we had an undetectable viral load, we reduced it to almost zero in the society, and the viral infections and the HIV infections should stop. This is how it looks like from the point of view of mathematics. I was part of that discussion, but life is greater than maths. People come and go. They don't want to. Some of them don't want to take drugs. There are many factors that will be preventing from leading that experiment to success. Last year, there was a detailed report, and you can see it in front of you, and there was an article in Science saying that this maximum coverage, and this was a huge study that lasted for about 5 years. There was an unprecedented number of patients, and we saw no success. Even if we treated all the patients in that area, we could not reduce the new infections. We cannot say that this maximum treatment approach can slow down the epidemic. All right, of course, in terms of our therapeutical practice, it's clear that we need to do that. The earlier we start, the better. We reduce the risk of a variety of different diseases, but that would be our next med talk immediately after our session, when we will be talking about treatment. But now we're more interested in epidemiology, and mathematics didn't work. We have a history of dealing with smallpox and cholera and plague. Here in Russia, Vladimir Havkin designed this vaccine, cholera vaccine, for the first time, and the plague vaccine. He used it in India actually for the first time, and back then it got clear that one can deal with those infections. They treated him as white god in India, and there's an institute of Bombay named after this gentleman, Vladimir Havkin. And it became clear that the use of vaccines may stop epidemics. Smallpox, Viktor Zhdanov was the first one who announced the vaccine had been there already, but they didn't believe us, but we did it in Iran, and that's how Russian scientists working in Iran led to a global anti-smallpox program, which was quite successful. It's all about science. Of course, community and education were important, but this very specific vaccine working against the microorganisms initiating those, starting those infections. I don't really like this slide. And here's the slide on eradicating HIV infection. And there are things that we don't know how to do. Yes, we can diagnose patients, but it's not visually clear. This disease has a latent period. We don't have a vaccine. Yes, we have some drugs that become more and more affordable in terms of the price, or huge work of international foundations, and like in Africa, the efforts of this particular organization helped millions of Africans to survive and get those drugs, and they trained a huge army of doctors. They came up with this industry of treating HIV from scratch, and that led to some serious success. Nevertheless, there's no vaccine. What can help us deal with the HIV infection? Of course, we need more fundamental studies to answer the question whether we can deal with HIV for good. It's development of treatment methods on prophylaxis or prevention. This is what we already mentioned in the morning. We need new methods, new pilot projects that would work. And of course, here we start with fundamental research, and then we'll be able to overcome HIV. We know quite a lot about HIV. That's probably one of the most studied, heavily studied virus on the planet. We know how it develops. We know its genome. We know its structure. We know how it replicates. We know the bio targets where the existing drugs work, and the new ones. We know which portions of the virus replication could be used, and there are other targets that could be used for patients. If we recall our very first patient, we didn't even know what to do with it. Then we got AZT, and it even helped our first patient. We remember those steps when we got those highly active antiretroviral therapy, which allowed many patients to survive that are still registered at our center and that got infected 20 years ago and more.
Today, the strategy that we have is to use combined medication, one pill that contains an antiviral complex, and a patient takes it once per day. I would like to remind those of you who have forgotten that before they had to receive a handful of pills several times per day. And of course, these medications were quite toxic. Today, we have broadened indication to treatment. If you remember, back in the days, we only prescribed treatment before a patient acquired immunodeficiency. Before, we used to believe that a patient should live for 5 to 7 years, and after we see a decrease in CD4 cells, we should prescribe a therapy. We believe that an indication was a level of 250 CD4s, then it increased to up to 500 CD4, and now even this level has increased for us to prescribe a therapy. And indeed, we have targets. Global healthcare has achieved some tremendous results when it comes to mother-to-child transmission. These are our data. We hit maximum low here. The figure is now at the level of less than 1%. We were the first in our country to work with pregnant women. We used new standards for our regimen. We worked with the author of the protocol, and she was absolutely impressed by how we used this protocol in Moscow, because we issued a decree of the Department for Healthcare, when actually every maternity department could prescribe this treatment to women, and it was done throughout Moscow. A similar strategy was adopted several years later by the US CDC, so they virtually copied our strategy of working with pregnant women in their guidelines. Today, pregnant women healthcare monitoring enabled us to achieve some impressive results. It is not only about medical factors, but it's also about time. Time is crucial here. Indeed, pregnant women do not like to be screened several times throughout their pregnancy. But this allows us to ensure that a child is born without HIV. Because sometimes, at early stages of pregnancy, there are no antibodies in a woman's body, but then later on, in the third trimester, unfortunately, we detect antibodies during screening, and in order to help such women, we have a protocol in place which is very effective. It works throughout the country, and I can tell you that in Moscow it yields significant results, which help us to ensure that mother-to-child transmission is curbed. But unfortunately, it is still impossible to cure people of HIV. We don't have a vaccine. And during our next Med Talk, we'll be speaking about a multi-resistant patient where we don't have virtually any medication that would be effective for the particular HIV strain. And together with Professor Rockstraw, we will be talking about how we can work with such patients, how we can manage such patients. Of course, those are unique cases, but even if we have one patient who is multi-resistant, we should help such a patient. And indeed, social implications of HIV are another crucial factor that we should consider. And the first question that we have to look into is whether a person actually wants to be tested for HIV, whether a person is ready to accept such a diagnosis. How do we treat drug users? According to data that we have, global data, they're not committed to following their ART treatment. In half of the cases, we cannot help such patients, even with the most state-of-the-art tools at our disposal. Indeed, another important question for us to consider is early treatment. Shock and kill strategy is another important factor for us to consider. Until this virus is not activated, activation of HIV response, such as through vaccines, but there are no vaccines to this day, decreasing immunoactivation of HIV. Leonard Margolis was speaking about that. How do we tackle this issue? Because you understand that virus needs high immunoactivity because it is a very conducive environment for a virus to thrive. Genotherapy is another popular field as of today, and many people are very hopeful about that because there have been some successes in oncology. And should these methods be transferred into fight against HIV, there may be some progress here as well. And the last point that we need to consider is stem cells transplantation. You know that 3 patients have been cured using this method, but many patients have died during transplantation, but nobody writes about that. We should all understand that selecting a donor is a very daunting and expensive task, and nobody is speaking about some mass transplantation just yet. What are the difficulties when it comes to developing an HIV vaccine? You know, it would be wrong for me to dwell on this topic because we have here Professor Shao, and he will be speaking about this in greater detail. He will be speaking about all tricks in this topic. I won't be dwelling on this. Probably the only thing that I would like to mention here is this table of the most significant vaccines that were tested throughout the globe. And many people put their hopes in the latest vaccine, a mosaic vaccine. The trials are still underway, and many believe that this vaccine does stand some chance to fight HIV. And I believe that during our Q&A session, we'll be speaking about that. But anyway, I believe Professor Shao will be speaking about vaccines in greater detail because he is a very prominent scientist. He is renowned worldwide, and it would be absolutely inappropriate for me to go deeper into detail about vaccines. So let us take immunotherapy. There are some medications here that are used, and I invite you all to a med talk that we are going to hold with Professor Rockstraw. We'll be speaking about multi-resistant patients and how these types of medications are used in them. Genotherapy is another option. There are various methods here that, of course, we will discuss a little bit later. But there are some surveys that are being carried out in this field as of today. And scientists, researchers have been seeing some serious results when testing this therapy on apes. But unfortunately, not everything that works for apes can be as effective for people. That is something that we should take into consideration. And that is an enigma to be solved. Yes, of course, we test our vaccines on apes. We have good results. But as soon as we go into phase 1 trial on people, nothing goes out right. There are various types of progressions when it comes to HIV. We have rapid or accelerated progression, typical progression, and there are so-called non-progressors. There are very few of them. There are such patients in every HIV center, and they're thoroughly studied for vaccine development. They are the foundation to look into how we can defeat HIV. So as far as data in Moscow is concerned, we have elite controllers. These are people who are HIV positive. They have been tested for HIV and they have been found HIV positive. And for a number of years, they have demonstrated an undetectable virus load. So their SD4 level is more than 500 cells. We also have HIV controllers who have HIV RNA of up to 500 copies, and their level of CD4 is more than 500 cells. And we have viremic controllers. These are people with viral load, and their HIV RNA is of up to 2,000 copies. And we have such patients at our center, and actually the next slide will be focused on them. Indeed, the criteria that we use to denominate our patients as these groups of patients are recognized globally. That is what our Moscow AIDS Center has to offer today. We have been monitoring our patients for many years now, and we have patients who for 19 years have been demonstrating an undetectable virus load. And that is a pretty impressive level and a pretty impressive time. So their CD4 level is more than 500 cells. We cannot detect HIV RNA in such patients. Speaking about elite controllers, we have such patients as well, and we believe that these are these patients who can offer us opportunities to study them more thoroughly and have some very useful findings. That is the work that we plan to carry out together with Moscow Institute for Technology. And we will use these patients for vaccine testing. That is my slide that you've already seen in the morning. This is a slide dedicated to ART, antiretroviral therapy. And now, of course, we are looking into long-active formulations of the integrase inhibitors. They should have been marketed this month, but one of the company was not endorsed by FDI. It doesn't mean that their formulation does not exist. It simply means that in the foreseeable future, they will once again send in their papers for approval, and they will have their medication on the market. So, indeed, long-acting formulations are important. We are speaking about injections once per month or even once per 3 months. And it is crucial for drug users, for instance, because it gives us an opportunity to treat them and to do so consistently and efficiently. And the number of patients who will have efficient treatment will increase. And it is not only true for drug users, but also for patients who are irritated at the necessity of taking a pill every day. So these regimens of taking long-active formulations will enhance the efficiency of that treatment. Well, I believe I'm speaking for way too long. So probably, if we speak about long-active formulations, there will be even more long-active ones that will be administered 1 or 2 times per year. Theoretically, it is possible to administer medications in such a way, and that is a path that we should discover for ourselves, and in the foreseeable future it will be very important for our patients. And it will be very important to decrease mortality among HIV-positive drug users. So that is what we should focus on in the foreseeable future. We will continue our scientific work to introduce new medications to the market. We will test these medications. We will conduct such work in our center, at our center in Moscow, and at other institutions in Moscow. Indeed, it is very important to ensure that we have not only foreign long-lasting formulations, but the ones that are developed here in Russia. We need innovative Russian medications that unfortunately do not exist as of today. We'll be discussing that in greater detail tomorrow. Today we only have copies of foreign medications. Today speakers were talking about that during the plenary session. It was important to introduce such copies, but the next target is to develop a homemade medication. Because today, Russian pharmaceuticals only develop previously developed medications. Our key focus will be on our work with our non-progressors. We will be studying them together with leading global HIV centers in order to achieve a victory over HIV. And I thank you for your attention.
Thank you for your interesting presentation. All right. What do we mean by comprehensive approach? I know how it works at FISTEC. First, we have genetic tests to assess how this disease develops in people, epigenetic changes, if possible. Those people who carry the virus, and then all of a sudden the disease starts developing in them. Perhaps there's some correlation due to change in the properties of cells or something else. Then you need an individual approach to therapy, and for that we'll have to use neural network. These are different labs. We have a separate CRISP lab, a bio computing for analyzing the data that we get, and we need to find those correlations to design better drugs and the vaccine. You understand that any illness is easier to prevent than to treat. But when we talk about HIV, it's really hard to design an efficient vaccine. But we have this unique opportunity to listen to an outstanding scientist, Professor Imin Shao, who will tell us about his achievement in developing HIV vaccine.
Again, thank you for your invitation. I enjoy being here very much. As our chairman coordinator mentioned, that HIV is a very difficult task to complete. If it's in English, they say a mission impossible. Why is that? I will just give a very brief history of the HIV vaccine, the phase 3 trial, what we call efficacy trial. We can see from here that there have been already several hundred clinical trials has been done. But the concluded phase 3 trials only 4. So we can revisit this 4. It's about 15 years ago, there's the first trial with the first generation vaccine, which is antibody-oriented vaccine with GP120, the membrane protein of the virus. We can see in the 3 years, the placebo and vaccine group infection rates are exactly the same. There's no efficacies. So, and I will just mention, we give the placebo and the vaccine double blind, and then the people will have a natural infection rates. So, and then we compare these two rates, is there changes. And after the first generation antibody vaccine failed, and the whole field moved to T-cell vaccine. They say antibody HIV is so variable, it escapes very fast, so we have no way to build a vaccine on antibody. So what they do is for T-cell. The T-cell, many trials have been conducted again. The Merck pharmaceutical company, they go the first. So they made adeno 5 vector as a T-cell vaccine, and we can see the trial results. To everybody's surprise, the two lines separated, but the vaccine group will have more infection compared with the placebo. And so we can two trials with the same results. The reason is not the vaccine caused infection, it's the vector they used are the wrong vector, because adeno5, 80% of people already got infected by adeno5. When they see this vector, before they see the vaccine, they have a very strong memory to the vector, cause a lot of cell amplification, providing more CD4 for the HIV infection. And NIH, the U.S. National Institute of Health, their national project, they call the Vaccine Apollo Plan, made by the President Clinton, 10-year plan. They happened to choose the same vector, adeno5, and they continued the trial with the adeno5 vector. They got the same results, and the vaccine group have 27 infection, placebo only 21. So they have to terminate this study. And the last trial, actually, what they do is use two vaccines at the same time. They pick up the same field vaccine, GP120, together with another vector, a pox virus vector, canary pox. So this vector, they prime first with the vector, pox virus, and they boosted with GP120. And then they get the second, another results. We can see this time the vaccine group has less infection compared with the placebo. So the efficacy is only 31% protection. This trial is done in Thailand, but the Thai epidemic is not high incidence. It's no use for Thailand, this vaccine. Now they are repeating the trial in South Africa. So China is still a developing country. When the big brothers all failed, so people say, why should we work on vaccine? Actually, we didn't think vaccine is easy. I have this chart I draw myself. I thought the vaccine is like a tale of two cities. The left column is the vaccine we already got, and the HIV is unfortunate fall in the right column. The already succeeded vaccine, they have one common feature: the pathogen stimulating protective immunity in natural infection. I use the example of Hep B vaccine. And actually, the whole science field didn't do much. They isolate the pathogen, they inactivated or attenuated the same pathogen, and they inject it to human body. And we don't know what happens after injection. And then but anyway, we rely on evolution, because on evolution, our immune system is stronger than the pathogen. And then, without knowing the exact what happened in the body, we already have a vaccine. So that is what we call CTA vaccine. It's a low-hanging fruit. So because the evolution is on our side. The HIV is just the opposite. So the pathogens, they do not stimulate enough protective immunity in natural infection. And then if we follow the same strategy, we do not have a vaccine. This is an example why HIV is different with the other column. For example, the hepatitis B vaccine, infection after exposure is a minority. So 80% of the people exposed to HPV vaccine, HPV virus, they can eliminate the virus without knowing. So we have like 600 million people in China have anti-hepatitis B antibody. They already cleared the virus, no virus. For HIV, we do not find a single person who has antibody without vaccine. And also disease after infection, mortality after disease. For Hep B, this is a minority. For HIV, is a majority. So that is a reason. So how can we do that with such a virus? At the very beginning, 20 years ago, we have followed these two strategies. I will go one by one. So first, we have to know the epidemic. As I mentioned in the morning, so about 30 years ago, I was in WHO. I'm the only virologist in the Global AIDS program. They asked me to build a viral research project for WHO. So I wrote a memo, suggested WHO to use the member country laboratory to build a network monitoring HIV variation. And this proposal is accepted. They asked me to organize the first meeting. So that's the first meeting the laboratory was built, Global Labs. And then I went back to China. I used the China CDC networks, doing the same thing. So we pick up mapping all the viruses in the country. We do every 7 years, 8 years national survey. And the first survey, we pick up the virus CRF07, the CN97001 virus as our vaccine strain. And this strain picking up is the right one because the scientist, Phil Berman, the corresponding author of this paper, he is the person who built the first vaccine, GP120, and also the last vaccine who has 31% protection. And he studied all the vaccine strains used for the clinical study, and he found our one is the best. So you see the yellow marked line, CN970. His virus can stimulate the best neutralizing antibody, should be used for the vaccine. And then for the immunogen, we also do differently with other groups. And we have two double strategy. One is to try to identify immune correlates and to choose the right antigen for the vaccine. Secondly, we thought we cannot still use the natural one. We have to redesign because evolution is not on our side. So first is we have to go out of the box because there's no successful HIV vaccine. We have to go out of HIV. Then we go to the virus. The HIV is a retrovirus family in the lentivirus genus. So in this group of lentivirus, 8 members, different animals. 25 years ago, I went there and to check which one, if there is a successful vaccine. At that time, I found there's one vaccine. It's for the horse. It's a horse equine infectious anemia virus. Vaccine was already built 30 years ago by Chinese scientists. And our academician, Professor Shen, from the Chinese Academy of Agriculture, he uses the traditional way of making vaccine, passing the virus strain in animal to increase the virulence, and then he culture the virus in tissue culture, in PBMC, in dermal cells, and over 200 passes, he got the vaccine. And he got the most funding 30 years ago, and then has no funding afterwards because he made the vaccine. And he came to me, and I looked for him, and we worked together for 20 years. He kept all the several hundred viral passages in good condition in his freezer. So we take it out using the modern technology, do the full-length gene sequencing, and we pick up how the virus is attenuated to become a vaccine. You see on top 100% killing animal, and then has no toxicity at all, become a vaccine. And we pick up the changes between the wild type and the vaccine strain, And we introduce those mutations from the animal virus to the human virus. And by this redesign, we can stimulate even better neutralizing antibody. And the last strategy we're using differently is we try to get a balanced immune response. Like I mentioned earlier, the first generation AIDS vaccine is for antibody, second generation is for T cell. So we want both. So how to get both? We have to use our strategy is using live vector. So all the past other groups, they don't use live vector. They use a dead viral or bacterial vector. We don't want to do the same mistake as Einstein criticized insanity, which is doing the same thing over and over again and expect different results. For example, the GP120 vaccine have made 50 times a clinical trial, and the non-replication vector have made 84 times a clinical trial. The replicating vector only 2. And that is by companies, Crystal Myers Squibb, and then they stop. I went there 24 years ago, asked why they stopped. They told me that because a commercial decision, because they don't want to do any vaccine, waste money, and they only go for antiviral drugs. So it's not a scientific decision. So we continue that work. So what we are choosing, I cannot move the slides. Okay. So we have to write to get a right live vector. Which one is safe? As clinical medicine or biology is experimental science. Unlike physics, mathematics, they are serious science. So what is useful or safe? We have to do trials. The largest clinical trial in history is smallpox. The smallpox vaccine in China in 960 AD, we already started using the live vector for smallpox vaccine. Actually, we are using on the mucosal immunization, you see, with the straw to blow to the nose of the children. And 800 years later, the British immunologist Dr. Jenner used the cowpox. So we choosing this vaccine. We choose what we call the Temple of the Heaven strain of the Chinese smallpox vaccine on the right, which is a tissue culture attenuated strain. To our surprise, Americans are still using the core skin derived vaccine. Even until this century, after September 11, they quickly immunized their 800,000 troops, and including Bush, the commander-in-chief, with the same vaccine in the old vaccine. So our vaccine strain, that is the New York of Health, Board of Health strain, so we choose our strain, 1010 strain. We put the 3 structured genes into the vector, and in the vector we can immunize in monkeys, do challenge study. We can get 87.5 protection for homologous challenge, and heterologous challenge we get 62% protection. So then we continued for clinical trial of this vaccine, and we did phase 1A, B, C, and 2A. We concluded already 4 clinical trials with the DNA vaccine priming, 3 injection, 1 injection with a live vector, and then we followed for a year. So the vaccine can cause 100% antibody response, about 2/3 T-cell response. So it's a balanced immune response. And it can also cause a similar level of the protective antibody like the RV144 31% protection strain. And because the vector strain, live vector, is very cheap, $1 you can already vaccinate 20 people, very cheap. So we try to see if we add a second boost with the same vector, it can get a much better results. We did a small trial, so we can see they can last very long, like smallpox. So we are currently doing a larger trial with 150 people with the second boost at 6 months, 9 months, 12 months. In about 1.5 years, we will get these results. And then we will move to phase 3 trials. So now more people are calling for using replicating vector, live vector for HIV vaccine study. Since we started at the very beginning with the live vector, so on the global list of using live vector, we are still leading. So we already concluded phase 2 trials. That made the US NIH came to us, asked for collaboration. They will provide us with a
В результате мы подписали меморандум о
GP145 vaccine, and together with our vector and DNA vaccine. So we signed an agreement with the NIH, with the China CDC, our Director General, Dr. George Gaofu, and we are working on that trial. So this is the current planned phase 3 trial of the world, as Dr. Mazel mentioned in his slides. It's RV144, now become a HVTN 702 trial in South Africa. They will get the result in about a year from 2 years. And there's a Harvard University and Johnson & Johnson pharmaceutical company combined trial. This trial is using adeno 26, which is few people got infected with adeno 26. That is, and then followed by our trial with Tiantan, Temple of the Heaven live vector, and then the last one will be Sino-US collaboration trial. So that's the future in the few years will have these 4 trial results. I show here is the lady is our first lady. She is the UN ambassador for AIDS and TB. So she paid a lot of efforts on promoting the public health. So when our president has a state visit to US, she went to the US university to see this trial and encourage the scientists to work together. Also, we can see the world target for HIV prevention has not been reached. According to the global plan, we should have less than 500,000 infections by the end of this year, but we cannot reach that as a history. You see the line followed will be still more than 1 million people got infected. So with the vaccine, the International AIDS Vaccine Initiative, they calculated, even it's only a 30% vaccine, they can reduce over 5 million infections by year 2030. If the vaccine efficacy goes to 50%, will be 70 million people be saved, or 70% efficacy will be 28 million people being saved. So that is the result of the vaccine. We are hoping we can reach there earlier than later. Thank you for your attention.
Thank you very much for an excellent talk. We'll switch back to Russian now. Во-первых, конечно, благодарю вас.
Thank you indeed for your talk. To my shame, I have to admit that smallpox vaccine was first developed in China, not in Europe. That was very interesting for me to learn. And of course, your talk, your report clearly shows us that the fight against HIV is not something that only private companies should be interested in. It is an issue that should be tackled at the state level. And as I learned it from your report, unfortunately, some companies just calculate how much money they will get, and based on that, they do their decision making, which is a wrong thing to do. States should be engaged in this work. And of course, your talk about vaccines and the efforts that China is taking up in this concern and that the US is taking up in this concern makes us very hopeful, and people will indeed thank you for your efforts and for your work. And I believe that we will support you in this endeavor. And how can we participate in that, actually? How can we play a part? Because we talked about drugs and medications, we talked about therapy, we talked about vaccines, and I know that our colleagues from the Immunology Institute from the city of Novosibirsk, they also worked on vaccines. Unfortunately, vaccines that were designed in our country, they never made it to phase 3 of the trials. Some of them made it to phase 2, but unfortunately, even these phase 2 trials, they did not receive any continuous work on them. But I know that in St. Petersburg, they developed a vaccine, and then it actually operated as a moderator of virus against antiretroviral therapy, which enabled basically to extract the virus from reservoirs. Unfortunately, today we find ourselves at the stage where these Russian efforts are somewhat in an impasse, though I'm not the one to judge. But virtually no work is underway now because there is no funding, and somehow there is no interest in continuing this work, which is very regrettable. But today we have some serious hopes to some joint efforts, some joint works with those who have already done some serious work on vaccines. And now we're working on some of these phase 3 protocols. We should have started to test this vaccine this year. Unfortunately, the preparation process proved to be longer than we planned when it comes to virus that circulates in our country. And as far as I understand, the talks at the Ministry of Healthcare are also pretty difficult, and without them they cannot start these trials. But there is this light in the end of the tunnel, and we do hope some progress this year. We do hope that the trials will start. Just like we hope that the trials for various ART options will be more robust, and I believe that will be a very good thing to do. But I would like to ask you a question. How many non-progressors do you have? How many elite controllers do you have, and do you use them to research HIV more thoroughly? Do you study these patients and how this cohort actually took part in your vaccine trials? Thank you.
Thank you. Actually, at the early times, we do have quite a few cohorts. We follow them. The longest one is a plasma donor cohort. We have followed them close to 20 years. They have it's hard to calculate because we did not start from acute infection stage. So, but we still found the added controllers of long-term survival. We have at least about 300 of them we follow. We use them for looking for the broad neutralizing antibody. We got several very good broad neutralizing antibodies. And also we try to study their the reason for their long-term survival. And we found that its innate immunity is pretty strong. And also some immune regulations. They have a pretty balanced immune response, do not have many inflammation response. And we try also to use their virus to look for if they can be used for vaccine. But that population is epidemic is already going down. It's very difficult to do a future trial. The future trial is the clade C vaccine and the clade AE. Those two vaccines in the MSM population, they have a very high incidence. We can use them for the future phase 3 test of the vaccines. So that's more or less my answer. But currently, because we have the strategy changed, we have to immediately treatment. We have no time to follow. But we kept all those samples in our liquid nitrogen, so we can still revisit them to try to fish out something. Yeah.
Вот здесь вот вопрос лечения.
But the issue of treating patients is indeed pretty sensible, and we will discuss it later on with Professor Rockstraw. But the strategy that we now have to manage HIV and HIV-positive patients somewhat contradicts our patient cohorts. If we are being honest and frank, yes, they're our patients, we monitor them, but according to our older standards, we should have started to treat them. But according to our today's standards, we would have already started to treat them. And we, by the way, have several children patients who are non-progressors. And according to the current guidelines, we actually violate the decree according to which we should treat them, but they do not require any treatment because their virus load is undetectable. Why do then we give them therapy? So that is a very tricky and sensible issue of a global scope. Indeed, treating HIV-positive people requires our most thorough attention when it comes to ethics. And it is a whole different topic, actually. And when we speak about controlling their antiretroviral therapy, whether they are committed to taking their pills and medications, it is also a very important issue. How do we do that? We have some time. We still have some time. If we have some questions, we will kindly ask Yelena to voice them. Dear colleagues, good afternoon. I collected the questions from our audience. And our first question goes to Professor Xiao. Does China carry out any research on long-acting formulations to treat HIV?
We do have several groups working on that. There's one new antiviral drug, which is a long-term, long-lasting fusion inhibitor. So it's made by China by pharmaceutical company. They just got licensed last year. Usually the injectable drug is used every daily, so they are using once per week. So they are working on a new formula once per month. They are also in collaboration with New York University group. They've tried to combine this fusion inhibitor with broad neutralizing antibody to make it like a quarterly used vaccine. But in addition to this group, there are not many. So antivirals are not very developed in China.
Not yet.
Спасибо большое за ответ.
Thank you very much for your answer. And the second question also goes to Professor Xiao. In your talk about vaccines, you spoke about their efficiency. So what criteria do you use to assess a vaccine's efficiency?
What we call efficacy. Efficacy is the protection rate. You do in placebo group, a vaccine group, and then you compare the natural infection rates. If the vaccine group, if the placebo group have 100 people got infected, the vaccine group has none. That is 100% efficacious vaccine. If the vaccine group have 50, that is a 50% efficacious vaccine.
Спасибо.
Thank you. The next question that we have, it is once again a question about long-acting formulations, long-acting medications. I believe that it is a question to all of you. Let us imagine a long-acting formulation is injected into a patient and it is an implant. How do we measure the dosage for a patient, or we use a universal dosage for everyone? And the second part of this question, what about drug-to-drug influence? How do we do that with long-acting formulations? Well, how do we do that is not a question for me, but rather for those who develop such drugs and who determine the contents of molecules in formulation. Indeed, if we work with well-known agents, prior to injection, we should use simple drugs, simple medications that would be fully compatible with long-acting formulations to make sure there are not any serious side effects. And only after that we should make an injections that will last for a month or so. So that is a question that should go primarily to those people who develop the formulation. Let me please chip in here. Of course, at our university, we have a lab that focuses on what we call nanorobots, probably not a self-revealing name, but it is not about the name actually. It is all about designing a tool, I believe I should call it a tool, that will administer a medication dose by dose by assessing the overall situation. Of course, we need to take up some great efforts to complete this work, but I believe that in the future, digitally, we will be enabled to inject long-lasting formulations depending on the blood works, depending on the feedback that we'll be getting from the patient's body, from the patient's system. Speaking about compatibility with other drugs, it is all called clinical pharmacology, but if we use neural networks or AI, name it whatever you want, I believe that this technology will enable us to track and to forecast possible compatibility or non-compatibility. And these technologies will help the doctor to see what kind of medications a patient should take.
Speaking, to monitor the patient's adherence taking drugs, normally it is self-reporting. They report how many drugs. And to be objectively monitor, they have this peer count. So the doctor goes to their nurse to go to his place and to count how many drugs left over. Or they even developed electronic, how many times he opened the bottle. But those are difficult. They can still trick. They can, just open it. So, and then you have the blood test, but it's very costly. Some scientists in U.S., UCSF, they developed the hair to test the drug level in the hair. Because the blood level is only for 2 days, and the hair is for 2 months, so you can cheaply monitor longer time.
Большое спасибо за такую интересную дискуссию.
Thank you very much for this interesting discussion. And in conclusion, a philosophical question to all experts. What do you think about the prospects of using extended use medications for long-acting formulations for pre-exposure situations? Pre-exposure products are more efficient than the vaccine. Okay, if we use long-acting formulations, perhaps if we use those extended use medications, it will make no sense to develop vaccine at all. Okay, let's say we have inexpensive products and you take it once a year. Theoretically, it's possible, and it will work as this protection from potential infection, HIV infection. I didn't tell you everything, but the pre-exposure prophylaxis is one of the critical factors that really slows down infection in the MSM group. The figures are amazing in New York, in LA, in the United States in general. These are very specific numbers in the UK. For the London cohort, we understand that the pre-exposure products are another important step in fighting the infection, reducing the number of newly infected in absolute terms. But we all understand that we do need the vaccine, of course. And our colleague from China can tell us more about that. But in terms of selling the drug, that will be a long-acting drug working as pre-exposure prophylaxis product. It's a good commercial project with, of course, it's more convenient for a healthy person than to take drugs every day, but that's just my opinion.
The philosophical question. I think generally speaking, there's no silver bullet for HIV control. So any measures has certain limit, including the long-lasting. This is one thinking I have. You have to combine a comprehensive approach, not only biomedical approach, but also social behavioral approach, education programs. And also vaccine and drugs, I think drugs are more emergency response, can be used to reduce the disease burden. But to really cure a disease, we never use drugs to cure a disease, but we do use vaccine. We eliminated smallpox from the earth. So that's the way the nature gave us the way of immune response to the pathogens. And anyway, as I said, anyway have limit. So the target is still the virus. And it's like even you have long injectable, you have vaccine, if the people do not behave, they have more sexual abuse and drug abuse. You just provide a bullet-proof jacket for them. It will maybe HIV disease burden goes down, but all the other sexual transmitted disease will be flood the world. So nobody wants to see that. I think it's just like the environmental protection. So how do we deal with that? We should control the pollution as the root of the disease, not to try to get some medicine. We can stand the pollution. Toxic water, food, no problem. We have long injectable drugs to prevent, to keep alive. But it's a miserable life.
Отлично, отлично. Да, философский вопрос, совершенно философский.
An entirely philosophical answer to a philosophical question. All people are different. HIV is a highly complicated condition. We need to have a variety of approaches in preventive medicine and classical medicine. People are unpredictable in all senses. Will we have a victory over HIV? That's our question of today's debate, and which we almost need to end. In conclusion, each one of us will have to say whether we will conquer HIV or not. I will start with, we saw examples of smallpox and cholera. There's this TV show in Russia when at the end they ask experts how probable is it that in 10 years a certain problem will be resolved. Well, with HIV, definitely not in 10 years, but we are definitely making some progress, that's for sure. It's quite obvious that to find the solution, we need more fundamental studies, and we need to use different approaches, and that's where the success is going to be. Currently, scientists are working hard all over the world. Even if we look at the number of studies, eventually it's going to give some results. And I do believe that Russian researchers are among those leaders who will be producing some results in genetic technology and the parity trials of new vaccines that will allow us to use them and manufacture them. That's quite promising. And of course, it's possible to design such drugs. How else can it be?
Just seeing that everything has both sides. For HIV, I saw the good thing is like when I was doing my PhD, HIV has not been found. My first major topic is tumor viruses. But during my study, the HIV was found, so I turned to HIV. The good thing for HIV research is that if you take this job, you will not get laid off. You know, you will always have the job. Like unlike Dr. Shen, the great scientist who made the first AIDS vaccine, he was the most funded scientist in China in the 1970s. But when he made the vaccine, he has no funding. So don't do the job too good. As in China, we are both communist countries, so you are the elder brothers for us. So we have one slogan we all remember. He said, the communists can only liberate themselves after they liberate the humankind. So Dr. Shen liberated the mankind on that animal virus. He liberated himself. He lost the job. But when even a young people, if you decide to take HIV research, I'm sure when you retire, you're still working on the same disease.
Завершающая ремарка сессии.
This was the final comment of the session. I'd like to thank our participants and have a good day.